Good morning, everybody. I'm delighted to be giving you the company presentation with a little bit more details on the complete analysis of the MERECA study that we just released this morning. I need to show you, of course, our disclaimer. But let me start by telling you what is MERECA, what is the study, what were the key objectives. So, as you know, MERECA is an exploratory study, is international, randomized and controlled, and it's also open label and is a Phase II clinical trial. We have enrolled 88 newly diagnosed intermediate and high-risk metastatic renal cancer patients. Based on a 2-to-1 randomization, the patients received either 2 intratumoral doses of ilixadencel before nephrectomy, that is the surgical removal of the tumor-affected kidney, and subsequent treatment with sunitinib or sunitinib therapy alone post-nephrectomy. The primary objectives of the study were to evaluate the median overall survival and 18 months overall survival rates. As a secondary objectives, we include the evaluation of safety and tolerability, very important, tumor response, and also exploratory, the CD8 plus T-cell infiltration in pre- and post-treatment kidney biopsies. It's important to explain that the exploratory study, the main value and objective are mainly twofold. One, to support the decision to proceed with the development of a product to pivotal phase. And we do this basically looking at all the data and giving it the same degree of importance. And of course, the second objective is to, using this data and these findings, design what will be then the next phase, what will be a pivotal study. So very important to really help us in defining what is the important data and what will be the next study, key objectives and endpoints. So let me give you the key results from this complete analysis that then I will go in more detail through this presentation. So an important result is that the survival as of July 2019 was 57%, what corresponds to 32 out of 56 patients that were treated with ilixadencel, and this compares with 43%, 13 out of 30. The median overall survival final value cannot yet be calculated in either group as the data is not mature enough. And I will show you when in a graph why this is the case. Very importantly, and as we already communicated, ilixadencel did not add toxicity when we combine it with sunitinib, and this confirms the favorable safety profile that we saw from past studies, and really continue to confirm and make ilixadencel very well suited for combination therapies. Additional results refer to tumor response. So the objective response rate, or ORR, is measured as the single best response, being a complete response or a partial response, within the 18-month follow-up. And this ORR was similar in the two groups, with 44% in the ilixadencel combination group and 48% in the sunitinib monotherapy group. But importantly, we need to look into the more details of this tumor response. And one of the exciting results, and very encouraging, was, as previously communicated, the number of complete responders that was higher in the ilixadencel combination group with 11%, 5 out of 45 patients, compared with 4%, 1 out of 25 patients in the sunitinib monotherapy group. Furthermore, and in more details regarding the tumor response, the ilixadencel combination group showed the following results. A longer median duration of response, 7.1 months versus 2.9 months in the sunitinib monotherapy group, and this is measured within the 18 months follow-up. We also observed a higher percentage of responses that were ongoing at the 18 months follow-up, 60%, 12 out of 20 patients in the ilixadencel combination group versus 33%, 4 out of 12 patients in the sunitinib monotherapy group. Another important result was the percentage of responders alive at the last patient contact. And we also have here a higher percentage. 85%, 17 out of 20 patients in the ilixadencel combination group versus 58, 7%, 7 out of 12 patients in the sunitinib group were again still alive at the last patient contact. These results definitely support the decision by the company to continue the development of ilixadencel as an immune primer in renal cell carcinoma and other solid tumors, and this is important for the benefit of cancer patients. Also support the planning of activities for the initiation of a Phase IIb, Phase III pivotal study in renal cell carcinoma, and also provides us with the opportunity to intensify the interactions with potential licensing partners. Let me show you again the MERECA design and timelines because these are important for you to understand the data. As I mentioned earlier, it is a randomized, controlled, and open-label Phase II study that was also stratified for high and intermediate risk, assessed by IMDC or ENC criteria. As you see there, the patients that all at diagnosis had metastasis were randomized in 30 patients to the control group where they didn't receive ilixadencel injections, and 58 of them to the ilixadencel group where they received 2 injections of ilixadencel at day 1 and 2 weeks later. And normally less than 8 weeks after the enrollment, the patients had the kidney affected by the tumor removed. And then, normally around 6 weeks, they initiated the treatment with sunitinib. And we followed these patients up to 18 months, and we did this for overall survival, progressed disease, and other endpoints. The study formally ended at the 18 months time point. That was, for the last patient, June this year. And after that, we will continue monitoring these patients for survival. And we will be doing that at 6 months interval. So let me give you some more detail about the patient distribution because it's important to understand the data. So we screened 117 patients. Of these screened patients, 88 patients were enrolled. And they were randomized in a 2-to-1 fashion, 88 to the ilixadencel treatment group and 30 to the sunitinib or control group. 2 patients never were treated with ilixadencel, so we were left with 56 patients in what we call the ilixadencel treatment group that will be compared with 30 patients randomly assigned to the sunitinib or control group. As normally happens in this type of studies, we lose patients, particularly in oncology, where patients or die or don't come for additional visits, remove their consent. So in our case, we lost 11 patients, 10 because they never received the sunitinib, and 1 patient because never received the scans, and we could not follow for tumor response. In the sunitinib group, this happens with 5 patients. So we were left for the purpose of looking at tumor response with 45 evaluable patients that were treated with both ilixadencel and sunitinib, and that we call the combo group, and 25 evaluable patients that were treated with sunitinib, that is the control group. So let me give you a little bit of the patient characteristics. As you know, in addition to being randomized to ilixadencel plus sunitinib or sunitinib alone, the groups were also stratified for intermediate and high-risk patients by the IMDC or ENC criteria. And a particular type of lesions, the sarcomatoid features, were also similarly distributed between the groups. And you see here the details that you can read. Now, in terms of the primary endpoints, let me start to give you the data. One of the primary or co-primary endpoints is median overall survival. So as I mentioned earlier, the survival as of July 2019 was 57% of patients alive in the ilixadencel treatment group versus 43% of patients in the sunitinib control group. The median overall survival final value, as I mentioned, cannot yet be calculated in either group as the data is not matured enough. And this is because of the fact that there are still patients alive in both groups, and this can change the final value. As previously communicated, the other co-primary endpoint, the 18 months overall survival rate, shows no difference between the groups. I'm happy to show you now a plotting of the survival. I think this is a format that you are used to see. And you see here the plotting for the 56 patients in the ilixadencel sunitinib group. The patients alive are colored in blue and the ones deceased in black. There were some patients that were alive at a moment where they withdrew consent, so we treat them as alive here. So this you see also the blue line vertical. This is accounting where you can contabilize the 18 months survival. So this is the picture of survival for all patients, the 56, as of July 2019. This is one precise point in time, and this is important, as I will explain you. When you look at the sunitinib group, the same way, you see in blue the patients alive, in black the deceased patients, and in this case, in the sunitinib group, one patient withdraw consent. You also see, and we marked in both graphs, there were the patients that never received sunitinib, and that of course will not be considered for assessing tumor response. Maybe it's easy when we put both graphs at the same time and gives you a more clear visual representation of where these patients are as of July 19th in terms of survival. Now, normally, we would show Kaplan-Meier curves, and Kaplan-Meier plots are projected cumulative survival by treatment group. And the reason why we normally don't show these graphs is because they are projections and they are continue to change as the time moves along. But, one of the things that we wanted to do was to give you full transparency on the data. But it's important that I spend some time explaining you to this graph, particularly for the ones that are not so used to analyze and see them. So what you see in terms of the lines is the cumulative survival for each group. In reddish, the sunitinib group, and in purple, the ilixadencel plus sunitinib. You see some small circles in both lines, and these refer to patients that at the time of follow-up are still alive. When you look at the bottom, this tells you for each group how many patients were alive at each time point. In this case, 0, 6, 12, 18 months. So for instance, you can see that at 18 months, the projection was for 33 patients to be alive in the combo group versus 19. Now, at 18 months, the data is mature. This is not going to change because we already passed for all patients this point. The next point that where we will then be fixing the data is when we will release you the next 6 months of survival that we expect to communicate to you in January. So you see there, these lines, please consider that these are just projections. They can change, but allows you to give you full transparency. As we proceed, and we do at each 6 months intervals, we will of course be providing you then what will be considered mature data that will not change. Now, let's look at tumor response. Tumor response is evaluated by CT scan or MRI at a central laboratory. So all the sites, the 28 sites, will send their images to a central laboratory that we will read these images in a blinded fashion. And the tumor response is measured by measuring the diameter of these metastases in what we call target lesions at predefined time points that I will show you. For MERECA, the tumor response starts to be measured from the start of treatment with sunitinib until the last visit at 18 months post-randomization follow-up or at disease progression if the patients progress earlier. And with this tumor response, we're going to be looking at objective response rate, or ORR. And ORR is the proportion of patients with partial or complete responses. These responses are measured by a standard called the RECIST 101 criteria, where you'll see complete responses, partial responses, stable disease, and progressive disease. In the next image is probably going to be more clear to explain you how these are classified. So this is the RECIST 101 criteria. So what we are measuring here is the change in diameter in these metastases from baseline to the point in time where we look at the lesions. That will be at different time points. And these are measured as a percentage. So you see in the scale the number 0, that is basically below, the tumor reduces the size, shrinks, and if it goes above, the tumor grows. Of course, the easiest one is the complete response. That basically, the tumor reduces in full or 100%. Then you have the group that is called the partial response. These are the lesions that are reducing in size from minus 30% to below 100%. Then we have the next group, the stable disease. And this group of patients can also have tumor shrinkage, but they will be below the 30% or below, plus 20%. So it's quite a diverse group because they can also have tumors growth, but they can also have tumors shrinking. But these are the criteria to define a stable disease. And of course, progressive disease, that is tumor growth more than 20% above what was the baseline. So let's look at responses. And this is important to evaluate really the synergistic effect of ilixadencel when combined with sunitinib. So I'm going to show you in more detail 4 specific aspects. Response rate, that is the single best overall response, being the complete response or a partial response, as achieved after the start of sunitinib at any point in time up to the 18-month follow-up. Duration of response is the period from first occurrence of response to progression or end of study, that is at 18 months. Ongoing response is if achieved response, the partial response or complete response, is still ongoing at the end of the study, 18 months. And then we also measure the responder survival, that is measured, of course, until July 2019, and we will continue to follow for 6 intervals. It's important here that you understand that for tumor responses, we are measuring within this gray box, start of sunitinib up to the end of study or 18 months post-randomization. So, you can see that the period of time from within this box ranges from 15 to 17 months. So let's start by looking at objective response rate. That is, as I mentioned to you, the single best response overall, being a complete response or partial response. This is just for illustration and shows you that although this patient had a partial response at the 12-week post-treatment with sunitinib scan, in reality the best response was a complete response that happened 12 weeks afterwards. So the complete response is considered the best response. So when we look at the data, you can see that ilixadencel had stronger responses. The objective response rate was similar between the groups, 44% and 48%. But in the ilixadencel combo group, we had stronger responses. 5 complete responses, or 11%, as compared to 1 complete response for the sunitinib control group, 4%. And you see there in the graph. Let's now look at duration of response. Because the response is just one point in time, you need to also measure for how long this response is going to last. So the duration of response is the period from first occurrence of response to the progression or end of study. Following the same example, you see there that the first response in reality was at 12 weeks post-start of sunitinib. There was, as I mentioned earlier, a complete response later on, but the duration of response starts when the partial response started. So the yellow line is going to be assessing the duration of response. All, again, as I mentioned, within this gray box. Next one is the, providing you with the data. And this is the part where you start seeing some of the differences and the impact of immunotherapies. Because immunotherapies are normally, ORR is not the best measure. You need to go into detail into the specifics of the ORR. And longer duration of response in our group is starting to show you some of those differences, where we had a longer duration of response, 7.1 months versus 2.9 months in the sunitinib control group, as you see in this slide. Now, the next one is, you have, of course, measure a duration of response, but this duration of response stops to be measured at the end of the study. So we also have to assess, is this duration of response that happened, after start sunitinib still there, maintained at the end of the study? So in this case, you see there was a complete response at 24-week scan. At the end of the study, the patient still had a complete response. So this means that this response maintained and is ongoing at the end of the study. So how does this then read in the MERECA study? You can also see that in the MERECA study, the ilixadencel combo group had more ongoing responses, 60% versus 33% in the sunitinib control group. Now, next one is, of course, also, and very important is, these patients maintain this response, but are they still alive? Of course, as you mentioned to you, we are only looking at the responders population in both groups. So here, you look, is the patient basically alive or not? At the last data point was July 2019. So what is the data? You can see also here that we have a higher percentage of responders still alive at the last patient contact, up to July 2019. In the ilixadencel group, 85% versus 58% in the sunitinib control group. And these responses, of course, and alive, include, in the case of ilixadencel and sunitinib, the 5 complete responses, while in the sunitinib control group, the complete response patient has died. Another piece of data is tumor infiltration. As I mentioned to you, this was an exploratory secondary endpoint. The tumor tissue from the surgically removed kidney tumors was available from post-ilixadencel treatment patients and in the non-treatment control patients. The analysis was performed on a tissue section from each tumor placed on a slide and immunostained with anti-CD8 antibodies. Tumor infiltration was then assessed on the entire slide by a new methodology, including a customized and validated computer algorithm software, to quantify the CD8 stained area as a percentage of the total tumor area. This analysis showed the median stain area of 1.08% in the ilixadencel group as compared to 0.84% in the untreated control group at the time of kidney surgery. The high variability of CD8 stain area in the tumors within the treatment groups between different samples taken from the same tumor and also within, for instance, the complete responders indicate that the intratumoral infiltration of CD8 plus T cells by itself, without considering CD8 plus T cell specificity, type and functionality, does not explain the systemic therapeutic impact of ilixadencel when combined with sunitinib. This is particularly important because a lot of more recent studies and publications really are showing that there are different types of CD8 plus T cells and only some of the phenotypes are more activated. Let me show you, because this is a new methodology, very different from the one that we use in the first study. And here you see the illustration of a 6% CD8 stained area. So, totally different way of measuring, but this is a more standard measure and the one more used these days because it reduces, of course, the introduction of human biases because the images are analyzed by software. But this gives you a little bit of a reference to images that we showed you in previous studies. So let me show you what I mentioned to you in terms of the high variability. So you see there for the patients in red, the distribution, each data point refers to what is the percentage of CD8 plus stain area as a percentage of total tumor per different treatment groups. So you see the high variability within each treatment group is, of course, as you can see there, slightly higher in the ilixadencel plus sunitinib group versus the non-treated group. But you see also, as an indication, for instance, the arrows in red refer to the complete responses, that you see that a lot of them are really very diverse, more on the low end. Of course, sunitinib only had one complete responders, but this shows you that really this very high variability within each treatment group. You see there really that the complete responders go across the different areas of stain, and as I will show you also, the difference in terms of stain area within samples of the same patient, because from each patient we took 2 samples. Okay, I think it's important now to show you, as an example, a case study from MERECA study, and we picked up a complete response because it's very illustrative of the dynamics of the study and how to measure these responses. So, the characteristics of the patient, aged 63, had a kidney tumor that was between 7 and 10 centimeters in the greatest dimension, limit to the kidney. Then the investigators selected the target lesions. Target lesions are the lesions that are going to be measured along the 18 months follow-up to see if they reduce, grow, or maintain the size, because, of course, the kidney is removed. In this case, this patient had 4 target lesions identified: lung, muscle, a lymph node, and another in the lung. And in addition to that, they also isolate the non-target lesions that you also need to follow up, but that normally are smaller and you cannot measure so precisely. In this case, the patient had 5 non-target lesions in the lung, in the retroperitoneum, and the abdominal wall. The tumor infiltration median was 12.1%. And here you can see, as an example, the high variability even within the same patient, that in 2 different areas of the tumor, the patient had a 19.7% stain area versus the total area of the tumor, and in the other sample, 4.5. So, patient was randomized. 4 weeks after the first injection of ilixadencel, the kidney was removed. And approximately 4 weeks after the surgery, the patient started treatment with sunitinib and had the phased imaging at baseline. In reality, very surprising for the heavy tumor load that this patient has, the complete response came very early, at the first scan at 6 weeks. Then at each consecutive scannings, basically this complete response maintained and was still complete response at the end of the study. So we measured the duration of response from the 6 weeks scan to the end of the study, and is again represented there by the yellow line. And as I mentioned to you, then we looked at the survival. In this case, this patient is still alive, and so far the overall survival is approximately 3.1 years. Another very important aspect, as you know, is the safety profile, particularly when drugs are used in combination. We basically saw a similar safety profile of ilixadencel combined with sunitinib as compared to sunitinib alone, as you see there in the graph. And what this means is that basically ilixadencel does not add toxicity to the already existing sunitinib adverse event profile. This confirms ilixadencel's excellent safety profile and ideally positions it to be used in combination treatments. Our positive feelings around the results from the MERECA study are probably better summarized in the two quotes from the lead investigator in the study, Professor Börje Lundberg, and from the highest recruited in the study, Dr. Magnus Lindskog. They see really that the stronger and more durable antitumor response linked to the tolerability and safety and the achievement of these complete responses are really what they, as practicing physicians, are striving to achieve in these patients. And they both were extremely excited with the complete responses, results that they very quickly emphasized that you rarely see in these type of advanced patients. But of course, I think it's also time to give you a little bit more update on the other programs in the company, as Immunicum is not just MERECA. So in the ilixadencel study, that as you know is recruiting patients and where we are assessing ilixadencel in combination with checkpoint inhibitors in the indications head and neck, lung, and stomach cancer, we are now in the phase 1B, where we are combining ilixadencel with the PD-1 inhibitor pembrolizumab or Keytruda. As you remember, the protocol initiates with a lower dose of cells, as is required by the FDA, the American Regulatory Authority. We enrolled the first patient beginning of February. In this, what we call staggered phase, you need to wait for the period of time, normally about 8 weeks, to follow up the first patient, and if there are no safety concerns, then a board approves to enroll the next patient, and the same way for the first 6. We are now in a period where we are reaching the point of moving from the first cohort, the 3 million plus 3 million cells, to the next cohort, that is to the use of 10 million cells, that, as you know, is the most usual dosage for ilixadencel. And we expect to communicate this move from cohorts very soon. When you look at the current landscape for the use of immunotherapies to treat solid tumors, you can see that we are positioning ilixadencel in a very good place. Of course, we have data in combination with the kinase inhibitors like sunitinib, but also other TKIs in renal cell carcinoma, hepatic cell carcinoma, and in GIST. We are also, of course, with the new study ILIAD to acquire data in combination with checkpoint inhibitors in non-small cell lung cancer, head and neck cancer, and gastric adenocarcinoma or gastroesophageal junction cancer. So this is really great because diversifies and amplifies the usage of ilixadencel in different types of tumors and with different classes of drugs. Now, as I mentioned to you, we are planning or starting to plan activities for what will be the next study. It will be a pivotal study in RCC. And this can be done by us or potentially with a partner. So when you look at the current treatment of renal cell carcinoma, you can see that the favorable risk patients are still treated with sunitinib because the physicians don't want to expose the patients to the high percentage of toxicities in the combinations of the CPIs plus the TKIs. Then the recent approves in first line in the intermediate and high risk patients is of course now pembrolizumab plus axitinib or avelumab plus axitinib. But a lot of patients don't respond or because of toxicity they need to change treatment. The current standard of care in second line is cabozantinib or nivolumab. This is the current treatment schedule for these type of patients. So comes the question, so what is the future for ilixadencel in this landscape? I think is quite bright because really we have a very diverse potential in terms of the usage of ilixadencel in the treatment of renal cell carcinoma. We can go in first line and basically be added on top of the already approved checkpoint inhibitor with the TKI. And this is, of course, a potential usage due to our excellent safety. Then, in second line, you can also combine with a checkpoint inhibitor. But also, again, expanding the flexibility in second line patients, the combination with a kinase inhibitor. And this can really be sunitinib or cabozantinib. So really, we have quite a lot of alternatives for the design of a pivotal study. And of course, this is part of the work that we are going to do in sharing the data with the authorities, discussing the design with experts, and of course, if we enter into some type of licensing collaboration with a potential partner. So, in summary, end to end, the encouraging survival that we have observed as of July 2019 and that we will continue to update you at 6 months interval. The stronger tumor response, the longer duration of response, the higher survival among responders, added to the excellent safety profile of ilixadencel, really support a decision by the company to continue the development of ilixadencel as an immune primer in renal cell carcinoma, but also other solid tumors for the benefit of cancer patients. Also supports the planning of activities for the initiation of a Phase IIb or Phase III pivotal study in RCC, being done by ourselves or with a partner. And also this great data provides us with the opportunity to intensify the interaction that we have been maintaining on a regular basis with potential licensing partners. So, I want to now thank you for all your patience to wait for these more detailed results. I hope now you share our optimism and how positive we feel about these results. And of course, we look forward to be interacting with you and answering any questions you may have. Have a good day.